Good evening and welcome to the new session of Pre-Med Pro. We are continuing with the chapter Human Excretion, and today we will continue from the mechanism of urine formation. Okay, it's a long topic consisting of PCT, DCT, the function of the Henle's loop, the function of the collecting duct, and the micturition complex. Okay, so let's start from the formation of urine. Okay, so write the heading: mechanism of urine formation. Mechanism of urine formation. See, it occurs in three steps. It occurs in three steps. Okay. Next, the ultrafiltration. It occurs in three steps. The steps are first step is ultrafiltration. Ultrafiltration. The second step is reabsorption. And the third step is tubular secretion. Tubular secretion. Okay, these are the three steps of urine formation. Now, take it as a note. What? About 1/5 blood pumped by each ventricle reaches kidney for ultrafiltration. Reaches the kidney for ultrafiltration. Reaches the kidney for ultrafiltration. Okay, now let me draw a small diagram of nephron. Okay, here let me draw it first. Okay, now here the Bowman's capsule region. Now here it is the afferent. It is afferent while it is efferent. Okay, now what happens? Here 1100 to 1200 ml of blood comes in. Okay, now here the ultrafiltration happens. They'll give the topic as ultrafiltration. So what is ultrafiltration? Ultrafiltration. So okay, ultrafiltration membrane, UF membrane, this is UF. Okay, the ultrafiltration membrane is composed of endothelium, that is of glomerulus. The second one consists of the basement, it's a non-cellular part, while the third one is the podocyte of visceral layer, the podocytes of visceral layer. Okay, now it is the podocytes of visceral layer. So what happens? Out of this 1100 to 1200 ml of blood, 1200 ml of blood, due to high pressure in the glomerulus, some portion gets filtered out. Some portion gets filtered out. So GFR, the glomerular filtration rate, amount of filtrate formed per unit time. So 125 ml per minute or 180 liter per day. This question comes in the exam. Now out of this, 1100 to 1200 ml of blood, due to high pressure in the glomerulus. So glomerulus has the high pressure. In the glomerulus, due to the high pressure in the glomerulus, some portions get filtered out. GFR, the glomerular filtration rate, it is the amount of filtrate formed per unit time. Right? Now it is given. Okay, now what is the ultrafiltration? What happens in the ultrafiltration? Ultrafiltration here, it is a passive process, so no active energy is required. It's a passive process. Okay, now only the plasma part gets filtered out, leaving a protein. So the plasma portion only gets filtered out, leaving the protein. Okay, this is the ultrafiltration. Now, the second part is reabsorption. Reabsorption. From the word, you know something is reabsorbed inside. Okay, from the word you can know that something is reabsorbed inside. Okay, the purpose is mostly to reabsorb, useful things from the filtrate in blood. Okay, so in filtrate in blood defines what? It is what is formed in, it is what is formed in Bowman's after ultrafiltration. In ultrafiltration. Okay, the basics. What's the basic is followed here? The basics: osmolarity. Defined in terms solute, solvent. Okay, now (100 grams of solute, 500 grams 1 liter H2O, 50 grams solute. Okay, now here what happens? The this is lead A, this is B. So osmolarity of A is more greater than B, because here it is 50 and here it is 100. Okay, 100 gram of solute and 50 gram of solute. So osmolarity of A is more larger than osmolarity of B. Okay, now come to the next important part, that is the proximal convoluted tubule. Proximal convoluted tubule, or the PCT. So PCT, in PCT, PCT, I am drawing a thing. It is very important. Just understand. Okay. Here, through here, it's a filtrate membrane. It's a filtrate membrane. Here, the filtrate. T-R-A-T-E. Filtrate. Okay. Now, what happens? Let it be the peritubular capillary. It is a peritubular capillary. It is a peritubular capillary. So, from the filtrate, here it is the interstitium, outer space. Okay, now what happens here is glucose goes inside the peritubular capillary. Amino acids goes inside. Sodium goes inside. Potassium goes inside. Chlorine goes inside. Bicarbonate goes inside. Water goes inside. And urea, a very small amount. To maintain our osmolarity is reabsorbed. Small amount is reabsorbed. Now a small amount of urea is generally reabsorbed to maintain the osmolarity. Okay, to 70 to 80% electrolytes and water is reabsorbed. Brush border epithelium, brush border cuboidal epithelium. It functions as to reabsorb the bicarbonate ions absorption. Now inside that note, take it as a note, what every solute has a threshold level in the blood. Beyond that, it cannot be reabsorbed. It can't be reabsorbed. Okay. Every solute in the blood has a specified threshold. Beyond that, it cannot be reabsorbed again. Okay. So that is called the maximum reabsorption. Okay. So here osmolarity remain same, that is isotonic. Okay, osmolarity remaining same, that is the isotonic. Now come to the loop of Henle. Give the heading loop of Henle. Okay, here H2O reabsorbs. Okay, 400, 500, 600, 800 ml. Urine formation 1200. Here the concentration happens. Here the fluid will reabsorb, thus the urine will get more concentrated. Okay. Now this is called the descending loop of Henle. This one. And The function of the descending loop of Henle, it is basically made up of simple epithelium, simple squamous epithelium. Okay. Permeable for, it is permeable. This question comes in the exam. Permeable for H2O, but less permeable for electrolytes. Okay. As the filtrate goes down through descending loop of Henle, it becomes hypertonic. Osmolarity goes up. Okay. Now, in the case of this ascending loop of Henle, what happens? It is made up of simple cuboidal epithelium. The descending loop of is made up of squamous epithelium, and it is simple cuboidal epithelium. Okay. Here, active reabsorption of electrolyte. Here the concentration of the urine happens. Okay. The thin ALOH, ascending loop of Henle, is made up of simple squamous epithelium. Here the reabsorption, take it as a note, reabsorption of sodium, reabsorption of chlorine, reabsorption of sodium, potassium selectively, and chlorine happens. Okay. Now, passively reabsorbed, ALOH, that is the ascending loop of Henle, is only permeable for electrolyte. As the, take it as a note, as the filtrate goes above ALOH, hypotonic osmolarity happens, and in case of ALOH, that is the ascending loop of Henle, minimum reabsorption. Okay. So today we have studied the PCT, the DCT, and the functions of the PCT and the DCT, how the urine gets concentrated. So in the next lecture, I will continue from the DCT, the functions of the DCT, the conditional reabsorption. Okay, and later then on that part, I will continue with the collecting duct and some important points. After that, the micturition reflex will follow. Okay, the mechanism of concentration of urine will follow. Okay, now I've just get in depth overview of the PCT and the DCT. The explanation of more the collecting duct, the PCT and the DCT will follow in the next lecture. Okay, till then, good night. Please subscribe this channel and like and share it with your friends. Thank you.